We are here with Maria Antonietta Tosches, who is an awardee of the John Kendrick Award, and we are happy to talk to you about your career and your science. So welcome and thank you for joining us.
Thank you.
So let's start perhaps from the EMBL, where I guess your career also started, or you have been anyway an alumnus, and we are doing this for the EMBL Alumni. So what was your experience at EMBL? When did you come? What did you do?
I came to EMBL for my PhD. So I came here, and the first impact was the pre-doc course, which was it was nice to just hear about what everyone at EMBL is doing. And my research was in the lab of Detlev Arendt, where I did, for the first time, work on Evo-Devo, evolutionary developmental biology. I had absolutely no idea about evolution. So the first lab meetings were very crazy for me because I couldn't understand what people were talking about when they were referring to all these different animal phyla that I had never heard of. And the idea behind my work was to study a worm, a marine annelid worm, to understand or infer principles of nervous system evolution. This is a marine worm that is very nicely positioned in the animal tree and enables us to make comparisons and to make inferences about the early evolution of nervous systems. And so that's what I did here.
So that's quite peculiar because, we as scientists, we are always used to these standard model systems, whereas, of course, this is an unusual model system. So was it a challenge to keep these things in the lab, or is, things that are completely established and you have like, tanks like with zebrafish for doing these things?
I have to say, because I was young, it was one of my first experiences in science. Before that, I did my master's thesis on frogs, and that was my experience. Everything felt very, very simple, and only afterwards I've realized all the work and the challenge of working on non-established models. So platinum arrays is easy to keep in the lab. We keep them into boxes and full of seawater. And so actually one has to make the seawater from purified water and salt. Or what Detlev Lab is doing is to bring seawater directly from the ocean. They have shipments, or at least when I was here they used to have shipments of seawater, because apparently the worms grow better in that water. Then they need to be fed every day with spinach, and then they have to be monitored every day because they mature, and once they reach sexual maturation, they breed, and after breeding they die. So if you don't catch them at the right time, you lose the worms and the embryos that come with them. So there are, of course, every model system has its own peculiarities, but the breeding, the culture were very well established when I came. One thing that was not established was the genetic tools. It was the very beginning. And I was lucky to be at the time where we got injection of embryos to work. And so we got the ability of injecting messenger RNAs, for example, to overexpress GFP or any other molecule we were interested in. So it was fun to be able to do this. And for example, one of my projects was to overexpress a GFP conjugated to histones, which then labels all the chromatin, and then you can follow cell divisions. And then we took these embryos under the microscope, and we were able to image development from following the development of the brain and all the cell divisions, and that was really exciting.
Very interesting. I mean, usually there's always challenging with these non-genetic model systems. Did you manage to do also editing? I mean, and probably those were the early days of CRISPR, but did you manage to modify genes more than putting reporters?
It was the early days of zinc fingers, which were the predecessors of CRISPR, and yes, I did that. And there's a sad story behind this, because I was studying a certain gene, a transcription factor, And I'd done morpholino experiments, which is another way to interfere with gene expression. And I wanted to confirm the phenotype with the zinc finger. So we got these zinc fingers. And it was years of work until we realized that the zinc finger was cutting in the first exon, as we wanted, but there was an alternative start codon. So the protein was still expressed because we were introducing frameshift mutations, but the protein was translated from the alternative start codon from the same exon, and that's where there was no phenotype. Oh. And because this isoform was enough to perform the function of this gene, it was only a few amino acids difference between the first canonical start codon and the alternative start codon. So that's, I think, one of the projects where it was a complete failure. But I learned a lot from it.
Yeah, it's a failure, but I mean, these are the usual things that happen. I mean, when you get surprising outcomes, then there is some knowledge, at least, now you have annotated much better the gene. Probably this is the hard way to do it, but sometimes that happens. So, and then you progress to your postdoctoral work. So where did you go? What did you do in that case?
So I went to Frankfurt, to the Max Planck Institute for Brain Research, and I worked in the lab of Gilles Laurent. And that was, so Gilles, my postdoc advisor, he was at Caltech before moving to Frankfurt. And at Caltech, he studied many species, but mostly insect olfactory neuroscience. And when he went to Frankfurt, he decided to switch to reptiles. So I was again in the situation of working on an unusual model system and learning things on the way. When I joined the lab was working on turtles. But then around the time I joined the lab, we had decided, or Gilles had decided to start also working on bearded dragons, which are some Australian lizards. They're called bearded dragons because when they get upset, their throat becomes black, so it looks like they have a big beard. Mm-hmm. And that's our initial idea was to make. Why are we talking only about failed experiments? So our initial idea was to make transgenic lizards, and we established breeding in these bearded dragons, and so it was fun to see vertebrate embryos, lizard embryos. So
you had quite some development there and challenges. I mean, with this zoo, which I guess it's a little bit of what happens with Evo-Devo, where you have, let's say, non-conventional systems. But in this case, why choosing such a complicated, if you will, model? So what was the advantage for the science that you wanted to do?
Yeah. So the science behind all of this was to study a cerebral cortex that is the cerebral cortex of reptiles, which is much simpler than the cerebral cortex of mammals. With the idea that being the cerebral cortex, cerebral cortices, homologous brain regions, there would be some commonalities in the organization and function of cortical circuits. And this is what motivated the choice of turtles first, and then they complemented with lizards, which are more robust for the kind of behavioral paradigms that you can run with them. And bearded dragons specifically because they have a big head. So my colleagues in Frankfurt were implanting probes to do recordings. And one of the outcomes of all this work was, for example, the discovery of sleep rhythms in bearded dragons, which are the same as our REM and non-REM sleep rhythms. And my contribution to that was to find out what's one of the critical brain regions that is involved in the generation of this rhythmicity in the alternation of REM and non-REM.
Oh, that's very fascinating. It's something that you have developed more in your lab. You're still keeping that, or you changed again when you established your lab?
I changed again. So 3 years ago. And when I started my lab at Columbia, I went back to amphibians, which is the system that I worked on in Italy as an undergraduate. But instead of frogs, I went to salamanders, specifically to a salamander, to a newt. Newts are salamanders. And we're working on this newt that is called a Spanish newt, or Pleurodeles balto. And one of the reasons was genetics. because you cannot get, or it's extremely hard to make transgenic reptiles. But instead, salamanders have external fertilization, and you can harvest fertilized embryos and inject them in the same way we were injecting platinum-raised embryos here at EMBL. So with this, we've been able to do to generate transgenic salamanders, and then to complement all the other kind of work that I started in Frankfurt with the genetics.
So you're still working on sleep, or you're tackling different function of the brain now?
Yeah, we're not working on sleep. We're more broadly interested in cellular diversity in the telencephalon, including the territory of the telencephalon that is homologous to our cerebral cortex, and trying to understand the diversity of neuron types and what they are doing in the behaving animals. So trying to bridge this comparative molecular level with the description of molecular description of neuron types to their functional role in brain circuitry.
Mm-hmm. And I mean, what do you make? I mean, there is this idea that the human cortex would be so much different from non-mammalian cortices. So how do you approach them? Are you looking into that, or how do you think working on salamanders would be predictive of such a different structure?
Yeah, it's true. There are obvious differences. A large part of the mammalian cortex is called neocortex because it's new in evolution. But however, many of these definitions have been based mostly on neuroanatomical comparisons, counting how many layers are in there. And what we've been able to do with using comparative single-cell RNA sequencing is to really look at the cell type level, defining cell types from their gene expression profile, and being able to compare cell types across species. Mm-hmm. And I would say from the work that I've done in Frankfurt and the work that now my lab has continued, there are two lessons. And one is that there are certain types of neurons and brain areas that are deeply conserved or extremely similar, going at least in the salamander, in reptiles, and in mammals. But the neocortex of mammals is indeed composed of glutamatergic neurons that have gene expression profiles that are unique to mammals, entirely new. So the most ancient parts relate to things like hippocampus, which is involved in memory and spatial navigation, or to the piriform cortex, which is another part of the brain that is involved in olfaction. But once you look at the neuron types that make up the 6 layers of the neocortex, you can see that indeed there has been a lot of cell type innovation in that part of the brain.
So, I mean, I guess, one part of the work would be having some references of what is known for humans or for mammals. So are you interacting a lot with these flagship projects that are running in EU and US, all this brain initiative? I mean, I know the US one is mostly based on Drosophila and model system, but do you have connection with those fields which are not necessarily geared towards evolution, but more on building atlases and understanding maybe function connected to behavior?
I mean, in some ways, yes. One connection has come up as in my role as an advisor and next generation leader at the Allen Institute for Brain Research, which is really pushing forward the field or the effort of mapping the cell types in the brain from a transcriptomics point of view. They've generated these extensive atlases, which of course build on the history of the institute that has started with in situ hybridization atlases, and now it's cell type atlases, to connect and interact with people that are working on the mammalian front, and they're doing actually very exciting work on comparing neuron types across mammals from many different mammalian species. So it's in US at the moment there are no initiatives or funding opportunities that are geared towards evolution, exactly as you said, but the scientific community is very open, and we're regularly in contact.
Good. So are you also, you were refer about modern technologies. I mean, you mentioned before in situ, which was like the old way to look at RNA sex, and so I was wondering, I mean, if you are involved in technology development. I mean, you seem to have tinkered previously in your career with new model systems, setup of this and that. Are you still developing in your lab technologies that might give a more sense or a better vista of the data?
No, because we're already busy with with getting things to work with. So I see our lab as more as a lab that is exploiting or transferring technologies that have been established in other contexts, for example, for the mouse system or the zebrafish system, and trying to implement them in non-canonical research organisms, because that's already quite a challenging and time-consuming step. Things that you think should work right away may take months or years, or even turn out to be impossible. One of the nice things about the salamander brain is that the brain is very small, like orders of magnitude smaller than the mouse brain. So one of the things that we've been able to do is to use whole brain in situ hybridization, not the classical one, but newest in situ tools with short probes. It's called the HCR, hybridization chain reaction, and that together with brain clearing and light sheet imaging. So now we can have three-dimensional reconstruction of gene expression for almost any gene, we want in the entire salamander brain, which has been really exciting, and especially good for my students that have been freed up from the task of slicing brains, which can be very, very annoying.
Sections and series of sections. So that's fascinating. I mean, one could say that you're doing a lot of work in porting technologies, and that's certainly a big thing. I mean, if all these are anyway cutting edge technologies. So I mean, I have like a general, maybe naive questions, but I mean the brain is always like a fascinating territory, and I guess when people talk about cortex, the thought is always coming to questions like self-awareness and development of desired function. I mean, higher functions in the brain. Do you think it's some of your research, although I mean these are animals that have maybe a more limited repertoire of these things, if any, but do you think that any of the stuff that you do, of the research that you will do, will open some of or help answering some of this question of higher function that are present in the human brain?
I hope so. I mean, I think the leap to human is a big one. I think their comparative work among mammals will be better suited. But definitely there are many questions, question marks on the evolution of cognition. And there's been a huge focus rightfully so, on mammalian systems and mouse as a model. But there are many things that that mice can do or mammals can do, and we don't know whether non-mammalian vertebrates can do. So definitely, the anatomical, molecular work that we're doing, circuit mapping work, will be the starting point to start then to look at also behavior, which it's the most challenging part. This is something we are already playing around with. But when you are in front of an animal out of its natural environment, and you come from all these mammalian biases in a way, it's even just designing behavioral experiments. It's extremely hard because you don't know from where to start. For example, my graduate student, she was trying to condition animals with food, and it never worked. And then we realized that these salamanders can go in some sort of hibernation state, and they can modulate their metabolism, and will go on many days without food, and they will not be seeking food actively. So all the stimuli and the paradigms that have been used for mammals need to be tuned. But I think once this is done, there is definitely a lot to find out about the evolution of cognition. I don't expect that there have been huge jumps, but probably some more gradual changes and in the behavioral and cognitive capacity of animals, and these changes still need to be mapped out.
Yeah, but I mean, I think one thing that I find very intriguing of, again, working with non-conventional model systems is that there's so much to learn, right? I mean, even things that maybe are not directly related could be tremendously interesting, and because with the systems that are normally used for biological research, now we have a certain coded set of behavioral assays, techniques that we're using. I mean, then the feeling, especially with behavior, is that you might be missing a lot of critical knowledge just because your assays are standardized, are custom to a certain organism. Whereas here is an example where maybe some side observation could be very interesting for, I don't know, some other fields, conservation, biology, or this kind of very unrelated fields. Yeah, one thing that I found interesting was that when you were talking about Platinaris, I mean, that there's so much diversity there. I mean, I bumped into Platinaris a bit for zoology, for some of the colleagues that are doing zoology. And of course, there are zoologists that are comparing that species with the nearby species, and the nearby species, which is, I was reading this thesis and they were discussing about the Gulf of Naples, and they were sampling Platinaris, and in that case they were using it for as a bioindicator of temperature of the seawater. And so it turns out that if you move like 500 meters away, you have a different kind of different species of Platinaris. And they are completely, absolutely ecologically completely different. I mean, it's a related species, but it doesn't die after mating. And they just live 1 year because there is, 32 degrees water, or I don't remember, 27 degrees water, and 400 meters, apart because there is 25 degrees water. Complete different ecological niche. So there's so much diversity.
Yeah, no, there's definitely a lot to explore from non-established, non-conventional animal models, and just the biology. So much to learn. Every time is an adventure when you start working on a new species, and you think you will get somewhere, and then suddenly there are interesting new questions that come up. So it's really challenging but fun at the same time.
So another question that I had was. What are your thoughts about starting a career in the US? Because you of course had previously worked in Europe, and you are starting as an assistant professor. You recently started as an assistant professor at Columbia. So what was the impact? I mean, I can't imagine the situation like that, where you also not only have to get accustomed to manage people, getting foundings, but actually do it in a completely different paradigm, if you will.
Completely different, yes. It's been enriching. I think as anything in science and life, being in new places, being in a new context with new colleagues in a different environment, it's very enriching. That said, I don't want to generalize from my experience, because I started my lab 6 months before the pandemic. So it was really a special time. And nevertheless, it took some time to adjust to the culture, but more than to the culture, because science is science everywhere, more to the language. I mean, I got there and people were talking about study sections, and I'm like, what are study sections? And everybody knows what is a study section, and I had no idea. It took me a while just to figure out how to read this RFAs. Now I speak the same language. It's funding opportunity announcements from NIH, for example, because I was not used to that. And then I got jealous of my students that from day one of their PhD, they get trained on how to write NIH grants. And I never had that training, for example. So that was a learning curve. I think the most shocking part of adjusting to the new system. But otherwise, it's been extremely enriching, especially because Columbia and New York are they're really rich in the diversity and kinds of science that is being done there.
So this has been a tremendous pleasure to talk to you, Maria Antonietta Tosches. I congratulate you on winning the John Kendrick Award. I hope that this will be something useful for your future, and I'm sure you are tremendously happy to have been awarded.
I'm incredibly honored to have been awarded the John Kendrick Award. It's something that is really special to me, because my time at EMBL was a special time of my life. The experience, the people that I met here, the research that I did here were shaping me as a scientist and as a citizen of the world. So I'm really excited to be here and to have received this award.
Thank you for talking with us, and good luck for the future.
Thank you.